Hi, everyone, and welcome to People Behind the Science. I'm your host, Dr. Marie McNeely, and I appreciate you joining me today for episode 703 with our guest, Dr. Kevan Shokat. Now, listeners, if you like this episode and you want to check out more stories from amazing people doing great science today, you can find more episodes of our show on Apple Podcasts, Google Podcasts, Spotify, or wherever else you like to listen. And today, Kevan has some excellent stories to share. So get ready to meet another one of our brilliant people behind the science after a short message from this episode's sponsor.
For 45 years, Lampire Biological Laboratories has been a key player in the life science industry. We are an international biotech company that produces biological reagents. For a complete listing of products, you can visit our website, lampire.com, or call 215-795-2838.
Every day, discoveries are made that will change our understanding of the world around us. Dr. Marie McNeely is here to bring you the brilliant minds who are making these discoveries so they can share their incredible stories and take you on an amazing journey. Welcome to People Behind the Science.
Hello, everyone, and welcome to People Behind the Science. Today, I am excited to be speaking with our guest, Dr. Kevan Shokat. So, Kevan, welcome to our show today. How are you?
Good. I'm great. Thank you, Marie. I'm looking forward to the discussion.
Well, Kevan, we are thrilled to have you with us today. And listeners, I'm excited to introduce you all to Kevan. Kevan is professor in the Department of Cellular and Molecular Pharmacology at the University of California, San Francisco, professor in the Department of Chemistry at UC Berkeley, and an investigator with the Howard Hughes Medical Institute. He received his bachelor's degree in chemistry from Reed College and his Ph.D. in organic chemistry from the University of California, Berkeley. Afterwards, Kevan received a Life Sciences Research Foundation fellowship to conduct postdoctoral research at Stanford University, and he served on the faculty at Princeton University before joining the faculty at his current institutions. And listeners, Kevan has received numerous awards and honors over the course of his career, including the 2023 Sjöberg Prize from the Royal Swedish Academy of Sciences, the 2023 National Academy of Sciences Award for Scientific Discovery, the 2023 Howard Vollum Award for Distinguished Accomplishments in Sciences and Technology from his alma mater, Reed College, the 2022 American Association for Cancer Research's Award for Outstanding Achievement in Chemistry and Cancer Research, and many others. He was also named a Pew Scholar in Biomedical Sciences, a Searle Scholar, a Cottrell Scholar, a Glaxo-Wellcome Scholar in Organic Chemistry, and an Alfred P. Sloan Research Fellow. In addition, he is an elected member of the National Academy of Sciences, a member of the National Academy of Medicine, and a fellow of the American Academy of Arts and Sciences. And today, Kevan, we want to get to know you both as a scientist, but also as a person. So can you start by telling us what you like to do when you're not doing science?
I like doing things with my family, my kids, biking with my friends, just getting out there away from email and looking at nature and getting some exercise and reading books about lately a lot of scientists, biographies and things like that.
Phenomenal. I think you're absolutely right there. Getting disconnected from the email and the inbox can be very helpful. And I love talking about books with all the people we have on this show. Do you have a favorite one that you can recommend for us today?
One that I just recently read is biography about Santiago Ramon y Cajal. That one was just really fantastic to learn about his life and what it was like in the early 20th century, late 19th century to do science, and so many commonalities with today, but so many differences.
Absolutely. It sounds like an excellent recommendation. We will add it to our website for our listeners. And we talked a little bit about your life outside of science, but I know describing what you do in science can be a challenge sometimes. So how do you approach this question, the big dreaded, what do you do for work kind of question?
I'd say that we have an approach that's interdisciplinary between chemistry and biology. And we try to look towards biology and medicine and find challenges that have not been tackled or places where discoveries might be found. And then we look in our chemical toolbox and find chemical approaches that might be the sort of missing piece. So that's kind of what we're always interested in, is how to apply chemistry to biology and how to do it in the most impactful, meaningful, interesting way to satisfy our curiosity.
I love it. And I think following your curiosity can be something that really keeps you motivated and inspired. But I'm one of these quote people. I always love getting quotes and sayings and just little inspirational things that I can keep in my mind as I go through my day. So, Kevan, do you have a favorite motivational quote or a mantra maybe that you use that's about science specifically or life in general that really motivates you?
Might be an odd one for a scientist, but I do ride a Peloton bike in the morning, and my Peloton instructor, she always says, "You did the hardest thing. You got on the bike." So I think part of science, the hardest thing is just get yourself into the room to hear the seminar and engage with it. And once you're in front of it, your curiosity just takes over and you start delving in. But those times in between those moments can be challenging. You sort of doubt yourself and things. So I don't know if I have a quote, except basically being in the room is the hardest thing.
Absolutely. I think getting started, taking that first step is huge. So if you find yourself dragging your feet, Kevan, what really helps push you to take that first step to get yourself to the room?
I guess just the many years of doing it and realizing that it might not seem like a fun thing to do, but then I've been so blown away so many times by what happens there that I remember, okay, I don't know what's going to happen, but the chances are something pretty special will happen. So get yourself to the room and listen to the talk and talk with the student, things like that.
Fantastic. I think the power of habit cannot be underestimated. So forming those good habits is great. So for you, when you think back about your scientific career, are there particular mentors or role models or people who might have inspired you along your journey?
Probably the most impactful person was my Ph.D. advisor, Peter Schultz, who was at that time a new professor at UC Berkeley in the chemistry department. And I still remember the moment I met him on an interview for graduate school. And he told me about the 3 projects that he was starting his lab on. And I had a rudimentary kind of picture of chemistry and biochemistry, but the things he told me that they were going to do, just my jaw was on the floor. And introducing the 21st amino acid into the genetic code, I was like, what are you talking about? It's not even possible. And what was great about it was you heard about this fantastic thing, and then he just was the master, has been, of breaking it down into very doable steps. And so no one step seemed impossible. Maybe it was improbable, but not impossible. So you got the feeling, wow, okay, I can do all these steps. And at the end, I will have this amazing thing. So I really take that approach to work in my lab. And ever since I've done science with him. So I love how you can break things down to doable chunks, but then it takes you to someplace completely different.
Definitely, where sort of the big picture is greater than the sum of all those small parts, those little pieces that you've been working on. So can you tell us a little bit more about your journey as a scientist? Maybe take us all the way back to the beginning, Kevan. Do you remember what first got you interested in science and started you on this path?
The only thing I remember, I wasn't a kid who played with chemistry sets or blew things up or things like that. I do remember early on a single question that just kept rolling around in my head, which was, what was the first thing that lived? I don't know if I knew about cells. I don't know what. I just was like, what was the first thing? So I was very curious about that. And then in high school, I got interested in chemistry and biology. The classes were less academic than maybe others were, but we did things like observe the candle burning and thought about how that worked. And so I was unconsciously, I guess, getting very good training at the fundamentals of the scientific method and observation and things. And then when I got to college, Reed College up in Portland, Oregon, I met some fantastic professors, including my intro professors, Tom Dunn and Phyllis Cozen, and they then launched me into the real science of it, periodic table, thermodynamics. And I just loved it. I just loved being able to take practical things and then convert it into a molecular picture. And I guess that's what I really like to do. Think about pressure volume, but then you think about heating up water and boiling, and you're just back and forth between the atomic and the visible.
Absolutely. What's going on at these tiny, tiny scales that makes this larger scale phenomenon that you can see? I think that's so cool that they were able to kind of bring the real life application to it to make it interesting and accessible for the high school and college audiences. So then can you tell us a little bit more about what your experience like was at Reed? Did you get involved actually in laboratory research at that point, or was it more being engaged in your classes?
At the beginning, it was classes. And what was really special, and I think is special about Reed, is that each intro class is taught as if everybody in the class is going to ultimately get their PhD in that topic. So it's given at a real depth. Calculus starts with number theory and proving that 0 + 1 is 1. Psychology is also similarly fundamental, and as is the humanities classes. So the first couple of years, I didn't work in labs. I just took the classes. And in the summers, I worked at my parents' print shop back here in the Bay Area. So I needed to sort of do that work instead of diving in for research. But in my junior year, I started to work in lab with Ron McClard, a biochemistry professor. And every month that I stayed there, I got more and more excited about research.
That's wonderful. So then can you tell us, Kevan, what were some of these pivotal moments, these key points that really took you from this college student getting excited about chemistry and biology to where you are today?
I think at Reed, kind of working on science that was actually kind of new and not just textbook, that kind of idea that you were working in the lab and doing an experiment and you could discover something that nobody else had ever discovered. So that fresh idea, that was really fun. And the more I delved into that, the more I liked that. So that's when I decided to go to graduate school. I was actually thinking about medical school or graduate school. And I think my senior year, I was applying to both MD, PhD programs and PhD programs. And the sort of decision was taken out of my hands because my current institution, UCSF, did not accept me to the MD, PhD program.
Oh, no. They probably have regrets.
It was leaning that way anyways to go to PhD, but it was funny in retrospect how that happens. And then also I met Pete Schultz. And that immediately swung me to do that. It was like an acceleration in that time.
I think sometimes these happen for the right reasons. So
yeah, I think there's so many times people think, I've got to get this thing I applied for. And I think in my career, I learned if you don't get that thing, that's actually a good thing because it wasn't a good fit. Maybe you didn't, the applicant didn't even realize it. But it's good to not go into a place that's a bad fit and let other things happen.
Absolutely. It keeps you opened up for other opportunities. And then you ultimately ended back up in the UC system, University of California, San Francisco, as well as your appointment there at UC Berkeley. So can you talk a little bit about how you found yourself to these positions as well as the Howard Hughes Medical Institute?
It was a long process from graduate school to starting your own lab. I would say that near the end of graduate school and all through graduate school, I always thought I would like to stay in an academic setting. And the things that were the friction between wanting to have a career in academics and following my curiosity were that sometimes your curiosity takes you on a little bit atypical path. And most academic institutions are extremely conservative and box checking. So if you don't take the path they took, you won't get the chance to have your own career. So I left my PhD in chemistry and did a postdoc in immunology at Stanford with Chris Goodenow. And that was such a big pivot that when I applied for my assistant professor jobs, back in chemistry departments or in medical schools, most committees didn't understand a person that had done chemistry and immunology.
They didn't know what to do with you. Yeah.
Yeah. It's funny. A couple of years after that, my friend and former colleague, Carolyn Bertozzi, who won the Nobel Prize this year, she also was a graduate student in chemistry at Berkeley and then did her postdoc in immunology here at UCSF. So we were shifted by a couple of years, but we both had that similar experience of people not quite understanding what kind of a scientist they were going to hire. In my situation, I basically had only one job offer out of all of my interviews and applications, and that was at Princeton. And that was really lucky because there were some really visionary professors there like Dan Kahn and Martin Semmelhack and Arne Levine, who actually were looking for somebody that bridged from chemistry to biology. So that was like the one place that I sort of fit in, and it turned out to be fantastic. It was great to start there, and I really, really enjoyed my time there. And then a few years later, I decided that my family was on the West Coast and we'd probably get to come back. And so my wife, Deborah, and I decided to move back, and our 3 kids came. So it was a big upheaval, but it was pretty early on. And now I've been at UCSF for about 23 years.
Absolutely. Well, I'm so glad you shared your story, Kevan, because I think a lot of people really feel lonely and isolated in this struggle to find that first faculty position. And it's really reassuring to hear that even really accomplished scientists like yourself kind of had trouble finding that place that was a good fit for you and your background and your interests.
Yeah, it's very easy to get your heart set on a certain university or a certain geographical area or a certain department. And it's really hard to explain to new postdocs how many things have to go right,
Right.
for a department to say, this is the person that we want. And so it's a real gift if your family is flexible, like mine was. My wife really left a great job here in San Francisco and found new jobs in Princeton, which was not easy. And having a supportive family is priceless. And I really appreciate all of that. There's so many things your family does that's important for your life and your career, but being flexible and things like that is a big ask.
Definitely. I think having that support network can make all the difference, because starting a faculty position and maintaining a successful lab over the years is not easy. And I know, Kevan, you've been doing amazing work in your research laboratory. Is there a particular project that you're working on right now that perhaps you're the most excited about and want to tell us a little bit more about today?
It's a pretty special time in the lab, actually, because we came through the pandemic, and a lot of things we were trying to do before the pandemic, they're the same projects, but somehow through the pandemic, because time was stretched out, we took more risks. We kept at things maybe a little longer than we would have. And a few things have really broken our way. So I don't know if the lab's ever had so many things going so well. Usually a lot of things are failing and you're trying to decide, shut it down or keep going. So the vision we have for the lab right now is we really want to get to something you really hesitate to even say out loud about cancer, which is a cure. And we really want to develop methods, molecules, approaches that can sort of tackle the really big challenges in cancer. And one of those that we've been at for about 15 years is trying to make a drug that would block the most common oncogene, KRAS. And that worked first for us back in 2013 against one particular mutant that's common in lung cancer. That's the KRAS G12C protein. And that kind of revealed a new pocket that we hadn't seen before. And now there are 2 approved drugs that work that way. And now the momentum in that field is just fantastic. And we've continued to participate and find drugs that target the other mutant alleles. And the chemistry that we're able to make work is something I thought would maybe happen in 2030, honestly.
Oh, wow.
But when you think that you'll work that long on it, you know it's an important problem, but you don't know if it's possible. I mean, if you have to work on something more than 5 years, it could be impossible or it could just be super, super hard.
Right.
But anyways, we really started to get some traction, and a lot of it is enabled by the clever chemistry that the postdocs, Xiang Zhang, who's now the professor at Berkeley, developed, and Chenheng Zhang and Johannes Morstein, and these chemists that have really taken new chemical strategies for these alleles, but also because of so many companies working on the target, they've developed much higher affinity molecules and shown us crystal structures. It's a huge effort that there's probably 15 companies at minimum, and there's probably between 50 and 100 scientists working at those companies on this particular target. And that's been in the last 5 years. So the amount of person years of science we've seen in the KRAS field is amazing, and it's really spreading, and everybody is benefiting, and hopefully ultimately the patients will benefit. So that's one side towards better therapies. And the other part is trying to leverage the immune system to attack tumors. So we have a lot of projects in that space in the lab, and we're trying to basically educate the immune system to find the tumors that we treat with the drugs. And we know from the last 10 years in cancer therapy, the checkpoint therapies that Jamalison and others have developed are breakthroughs. But we really need to sort of allow patients to see both a checkpoint therapy and a targeted therapy that kind of work in synergy. And then we can really have a much better handle on tumors. So I'm excited about both the immune side and the oncogene inhibition side. And the last piece is trying to leverage how cells respond to tumors using the tumor suppressor system. And we're trying to rescue tumor suppressors that are lost, like for P53. So we're trying a 3-armed strategy, and I can see the momentum. We've made a lot of progress.
Definitely. Well, Kevan, I think your lab and your collaborators, your colleagues are doing remarkable work here. And I think you're absolutely right. This sense that as the rate of discovery is accelerating, you're starting to get that momentum. And I think that is one of the most exciting things about science, when you start to see all these pieces falling in place. But I know, as you alluded to, it's not always like this. You don't always have these exciting, thrilling successes to celebrate in the lab. And oftentimes it's just like when everything goes right, everything tends to go wrong at the same time, I think. So
yeah.
Do you have a time in your experience or your career where everything was going wrong that you can tell us about, or you had a major failure that you had to work through?
I guess in the lab, we haven't had a stretch where everything was going wrong. Usually a couple projects are working and a lot of projects may be hitting roadblocks. So when you have a lab, you generally have multiple projects. So usually things don't line up that way. I think what gave me the ability to handle the difficulties was in graduate school, the first 4 years of my career, I was working hard, but none of the projects were reaching that key step of, okay, we have something and this is going to lead to publication. And it was funny, 4 years went by and it never bothered me. I guess I just kept going. I kept being interested. And it was at that time my advisor started to worry. It's like, Kevan, you're working hard, but nothing is working. What is going on? And I was like, wow, okay, you're right.
Just put it all out. Yeah.
Yeah. And it was a really good lesson for me, because I think it taught me that I'm an optimist. So I always thought the experiments I was going to do, the next were going to work. And that's the key thing in experimental science, is having a confidence that the next experiment will work. But then it taught me I do need to step back sometimes and think strategically. And around that time, one of the postdocs in Pete's lab, Christian Leumann, a professor now in Switzerland, he was leaving back to Switzerland and he had a shorter time in the lab. So he had started some projects, but they were not quite finished. So Pete gave me those projects to work on, and every one of those worked.
Oh, wow.
It was fantastic. I just started up, did the next experiments, and they were the things that really, we tweaked some of what the original goals were and things like that. But it really taught me then, and I use that now in the lab, that sometimes one of my graduate students has been just working so hard on something, and then a postdoc is about to finish and a second project of theirs needs to be picked up. And I try to be very strategic about who picks up that project. And that has been great. It's a way to short circuit a lot of time and give people the experience of all stages of a project.
Absolutely. So I have to ask, did you ever have that nagging question in the back of your mind during those 4 years? Is it too late to reapply to medical school?
No, I got thoroughly rejected, so I was not worried about it.
Okay, good. Well, Kevan, I'm glad to hear that this story of struggle had a happy ending. You were able to work through that PhD, and you've had so much success since then. So do you have a favorite success story you'd like to share?
Wow, I guess it's probably just the first project in the lab that was engineering kinases. I was really interested in how protein kinases work and which ones are responsible for which aspect of biology. And so I started the lab around an idea to engineer one kinase to accept a sort of non-natural substrate. and that would give us a handle on one kinase at a time. And I know when my first graduate student, Yi Liu, started to engineer the protein kinase and find the mutation to put in, and then my postdoc, Kavita Shah, had synthesized the analogs, and there was a match between the unnatural analog and the mutation. And we saw the activity come back. It was just like, wow. I still remember the night that the repeat of the experiment happened, not the first time. The first time happened, and then for 2 months, we couldn't repeat it.
Oh, no.
And so it was so nerve-wracking. And then E and I looked at the assimilation counter and we saw the signal in the right part of the trace. And it was a real special moment. I sort of knew we had it, and I knew the lab was like on solid footing. And before that, it was probably too risky of a project to work on. But I thought I have my one chance to do what I want to do, and I should do that. And a number of people asked, what's your backup and what's a more safe project? And turns out the semi-backup projects didn't work. So
oh, no.
they were harder than we thought. We thought they'd be easy, but they were hard.
That just means you were aiming high. I like it.
Yeah.
Well, Kevan, how did you celebrate then? It sounds like this was perhaps late at night working in the lab. You see finally that you were able to replicate this majorly important experiment.
I think we had some kind of a little party in the lab. I don't remember afterwards. I mostly remember the 2 months of going down into the basement and seeing no activity. And it turned out that what turned the experiment around was that it was the summer and the Summer Olympics were going on. And after 2 months of trying to carry out the experiment all in one day, so we would start early in the morning and we'd end around 2 a.m. Instead of doing that, my student, said, hey, Kevan, I want to put the cells in the fridge tonight so that I can go watch the diving competition, because the Chinese diver, I think her name's Li Na, that was her first Olympics. And he said, I really want to watch that. And I said, yeah, no problem. We're just like 2 months of every night not working. So put the cells in the fridge. Then the next morning, he started again. And that night, the experiment worked. And we were like, what? The only difference was we put the cells in the refrigerator. And it took us a while to believe that that was the difference, but we definitely realized that. And it turned out that the protein we were working on, SARC, needed to be folded by the chaperone system of the E. coli. And we were taking it through too fast and we weren't giving it that time. And in the fridge, it allowed the time to happen. So I always think of that Olympics as the real breakthrough and the importance of luck.
Sounds like serendipity struck again.
Oh, it's always the best.
Well, I think celebrating these amazing wins in the laboratory is definitely a fantastic part about being a scientist. And one of the other things that I love to celebrate are the opportunities that scientists get to travel around the world to work with colleagues, to go to meetings, to train. So, Kevan, do you have a favorite place that your science has taken you?
I think one of the nicest places I went and the most enjoyable trips was to Italy, to Sicily. And this is maybe 15 years ago. Went to a mountaintop city called Erice with my colleague, Brian Shokat. And it was a structural biology kind of meeting. And it was so otherworldly walking along the cobblestones at night in the fog on the top of this mountain. So the views from there were great and just a lot of great time to meet with people. And I met a few professors whose students then joined my lab as postdocs, including a very special postdoc, Daniel Rao, who really brought crystallography to our lab. And now we do our own crystallography. So the serendipity of being at a great place, meeting somebody, and then having follow-up output from that. Without that meeting and that city, I probably would have never met Daniel and would have maybe not done crystallography as much. And that led us to do the crystallography on our RAS project, which the two people on that project, John Ostrom and Wolf Peters, they benefited because 10 years before that, we had been doing a lot of crystallography. And so when we had our lead for the RAS project, Wolf was able to really tackle that and solve the structure, which showed us the pocket on RAS that I mentioned before. So it's just really fortunate to have great people in the lab. They always bring something new. And then the lab is a lot more equipped to do more difficult things and do them within the lab. That makes it so much more fun because we get to see it.
Yeah, absolutely. And I love this story because it really highlights the importance, I think, of scientific conferences when you've got great scenery, wonderful food, good conversation, excellent science, and bringing everybody together to have these, again, serendipitous meetings that may lead to fruitful collaborations or training environments, and introducing entirely new methods to your lab, I think, is phenomenal.
Yeah, science is always fresh. It's always new.
Keeps you on your toes. I love it.
It's fantastic. Yeah.
And I know there are some stereotypes, Kevan, about how scientists are portrayed in the media and what life as a scientist is like. And we try to break these on our show by showcasing these human stories in science, because I think so many people that I've met who are scientists go against these stereotypes. They're incredibly funny, creative, amazing people, not these lonely, grumpy scientists working in a white coat in a lab. So do you have a story of your own of a quirky tradition, or maybe just a funny science memory that really goes against the stereotypes that I think a lot of people still have?
I think that scientists, when you see them, they're not really grumpy so much as they're engrossed in something. And that when you walk by them, they might not engage. And I know for myself, it's not because I'm not happy or wanting to engage with somebody, but I'm just coming out of my office, trying to go get some water, and I'm still trying to puzzle something out. And the reason I say I think it's important is the small thing is during the pandemic, there were a lot of times when it was pretty quiet around and you'd see somebody in the hallway and it just seemed crazy to walk by them and not say hello and smile and ask them what's going on, because they were the first person you saw for that whole month.
Right.
And I've tried to carry that forward. And I really try when I walk around to smile and look at people. And I've tried to sort of break what's going on in my head and try to snap out of that and talk to everybody in the building, security, facilities people, the custodian people. And it's so important. Everybody's so important to the whole endeavor. And I think if we don't acknowledge people in our day-to-day interactions, it really makes for an unhappy environment. And we really, really need to break that. I think it's a small thing, but the last 2 years, I've made a conscious effort, and it's been very nice. I feel a lot better when I walk around.
Absolutely. I'm really glad you shared this, because I think you're absolutely right. The pandemic helped us all really appreciate that these small interactions, these small connections with people that you sort of meet throughout your day, just the impact of them not being there, and then just starting to be a little bit more intentional about your interactions with people can make a big difference, not just for you, but for everyone that you're interacting with who might be having a rough day, and then just getting that smile and that hello can kind of turn things around for them.
Yeah, totally.
Well, Kevan, we talked about some of the phenomenal work that you're doing in your lab, but I know oftentimes things stand in the way, whether it's time, funding, feasibility, just some of these roadblocks that you run into in your project. So if we took away the barriers that you normally face, is there one research question or problem that you would want to answer most?
I do have to say I'm very, very fortunate. I feel like we've been able to pursue any project we wanted, which is amazing to be able to say. It's just very, very fortunate. The Howard Hughes funding that the lab has and the collaborations and grants that we've had over the years. Even when money was tight, I always felt like we're going to do it. If it's a good enough idea, we should do it, and we'll work out the funds later. So I kind of don't work under the premise that something is holding us back. I think what holds us back is our ideas and how creative we can be. But the one thing I think is missing, it's not really our area, so it's not exactly answering your question, but I really wish we had a better readout of the effect of our drugs on cancer patients' tumors in real time. We try drugs in the patients we think will respond, but so often we just measure their tumor size, and there's so much molecularly going on. If we could have a more real-time readout so that we had a feeling of whether the drug was working the way we wanted and we could keep them on the drug, or if it was not working, we could take them off the drug and get them on a better drug, I think that would be transforming. And the next thing is getting the right drug to the right patient, independent of healthcare system or health inequities that we know are rampant. That just breaks my heart that a patient we know has a tumor type that could benefit, and the system at many levels fails them. Breaks my heart.
Yeah, these are the kind of things, the kind of questions and problems that sort of keep you up at night as a scientist. Just the idea of what can we do that will have the most real impact on patients, whether that's understanding real time and being able to switch therapies kind of on the fly and making sure that the people who need therapy most are able to access it. I think those are huge problems that we've still got quite a ways to go in working on.
Yeah.
Well, we've covered a lot of ground today, Kevan. We'd love to end with advice. So do you have a piece of advice that somebody gave you at some point in your career that really helped you that you can pay forward for all of our listeners today?
I really think the best advice is follow your gut. Your gut and your intuition is the main engine for discovery. And it's different in everybody, which is what gives us the diversity of scientific directions. So when you're in a seminar and your gut says, this is important, I should move in that direction. Or if it says, something's weird, I don't understand this and something doesn't make sense, follow that and dig into it. It's so incredible how intuition is sometimes at the moment hard to break down into a most reductionist picture, but it has this power of direction that is phenomenal.
Absolutely wonderful advice for our listeners. Follow your gut, follow your intuition. Kevan, is there any other last note of inspiration you want to leave everyone with at the end of our conversation?
It's another small thing, but I do think it's so important in science, and that is to challenge and ask questions. And so many seminars and group meetings go by where people don't ask questions. And that's such a missed opportunity because that's a chance for you to nourish your curiosity. And it also helps the speaker question their own assumptions. And I wish there were seminars are now an hour. I wish they were half an hour with half an hour questions, because that's where we really communicate and kind of find what the missing pieces are. So if you're a student, force yourself to ask a question. And it's nerve-wracking. I get nervous in a seminar when I raise my hand and ask a question. Absolutely. My heart rate goes up. Maybe you can say, I may have misheard you, or maybe you said this and I didn't hear it, or this is a really simple question, which is the killer.
Beware when you hear that. Yeah.
Yeah, beware. As a speaker, you know the dart is coming right at you. But it's a really important thing in science. If you thought of it, other people in the room are thinking it, and be the one that asks the question.
Well, listeners, definitely keep asking questions. Fuel your curiosity. Kevan, if our listeners want to learn more, what's the best way for them to do so?
I think our website at UCSF, which is shokatlab.ucsf.edu, shokatlab.ucsf.edu. Excellent.
Listeners, definitely check out that website. And Kevan, thank you so much for your time today.
Thank you, Marie. This was a great discussion, but time went by. It was fast.
It did. Well, it's been such a pleasure, Kevan. Thank you again so much for joining us on the show. And listeners, thank you for joining us as well. We'll see you next time on another episode of People Behind the Science.